Değerli katılımcılar, şimdi kongremizdeki sıradaki sunumumuz İngiltere Genomics Kanser Bilim Direktörü olan Dr. Alona Sosinsky tarafından yapılacak. Dr. Alona Sosinsky, İsrail'deki Weizmann Bilim Enstitüsü'nden biyoinformatik ve moleküler biyoloji alanında doktora derecesini almıştır. 2007 yılında Londra Üniversitesi Birkbeck College'da biyoinformatik alanında öğretim üyesi olarak görev almış ve çalışmalarının ana odağı dizi analizi, gen düzenleme ağları ve bunların embriyonik gelişimine uygulamaları olmuştur. Imperial College London'da biyoinformatik alanında çalışmalar yapmıştır. 2015 yılında Genomics İngiltere'de kanser analizi şefi olarak başlamıştır. 2021 yılından itibaren Genomics İngiltere'de kanser bilimsel direktörü olarak görev yapmakta ve genomik veri bilimciler ekibini yönetmektedir. Dr. Sosinsky'nin çalışma alanı, klinik genom dizlemenin kanser hastaları için maksimum faydalarına yönelik stratejiler geliştirilmesi, genomik ve klinik verilerin analizi üzerinedir. Dr. Alona Sosinsky şimdi sizlere İngiltere'de kanser hastalarında tüm genom diziliminin klinik uygulamaya entegrasyonu sunumunu yapacaktır.
Thank you for introduction. I hope it was nice. Thank you for inviting me to the beautiful city of Istanbul. It's my first visit. Let's start. And yeah, big congratulations with launching your genome program. I hope you will learn a lot from those who started early, and in 10 years, I'm saying 10 years because Genomics England is celebrating now its 10th year anniversary. So yeah, I hope in 10 years you will be well ahead. I will start from the history of Genomics England. It was launched 10 years ago. I've been around for the last 8 years, and find it's most challenging and most satisfying job in my career. Project 100,000 Genome Project that we probably still most known for was announced in 2012 by our then Prime Minister, David Cameron. It was launched 10 years ago, and it's co-occur with NHS. It's our National Health System 65th anniversary. It's now 75th, and it's now 10 years of Genomics England. 100,000 genomes were delivered within 5 years by the end of 2018. Actually, at the end, we over-recruited, so it's turned to be like 112,000 genome. Finally, in our research environment, we'll talk about it later. So it's take quite some time to build platform to ensure recruitment of the patient, build clinical pathways. The last couple of years, recruitment raised dramatically. This is why we over-recruited. And then next year, long-term plan from our National Health System was released, and it's talked about what we call now a patient genomic record. This is a lifetime record, similar to what Qatar colleagues were presenting. So it ensures that patient lifetime data, clinical data, genomic data is stored in a single place and allows research development of new drugs, refinement of treatment pathways. And of course, Genomics England responded to the COVID epidemic. We sequenced population of mild and severe patients, and we already had a big cohort of healthy controls in our research environment, and paper on markers of severe response was published a few years ago. The last but not least is the announcement in October of 2021, our newborn sequencing program. So it's kind of upside down if you compare Qatar genome study and genome program in UK. We started from 100,000 genome that was focused on patients with rare diseases and cancer. So we moved from this disease space into a screening program of the newborns. We start sequencing first newborns in several months. I think it's going to be more than 100,000 newborns screened as a first phase, and then reassessment of outcomes and hopefully broader enrollment. So that was a quick history of the last 10 years. So I will start from more recent events first. So once we completed sequencing of the 100,000 genomes, NHS launched genomic medicine services. We are not part of the NHS, but we partner with them. And then at the second part of the talk, I will talk about 100,000 genomes project and what is available for the research in our research environment. So genomic medicine services is a full path from clinical sample to release of results. We have now NHS test directory. You can just Google for the NHS test directory and you will see what it looks like. So this is defined the list of clinical tests that can be done for patients with rare diseases, for patients with cancer. This is not just whole genome sequencing, this is much broader range of clinical tests. And then this test directory is delivered through 7 genomic lab hubs. They collect samples, they run all non-WGS testing, they prepare clinical reports, and then this clinical, of course, sequencing data goes through the analysis, and then it's discussed on the clinical interpretation and discussion of results is happening on the Genomic Tumor Advisory Board, which are local to the health trust. And this is very multidisciplinary teams. It includes treating clinicians, oncologists, pediatricians, hematologists, also pathologists who review sample histology and make diagnosis, as well as genomic counselors. So this is a bit more details about our National Genomic Test Directory. As I said, you can Google it. At the moment, this is just Excel spreadsheet that tests all the genomic tests which are commissioned by our National Health Services systems. It describes technology by which they are delivered and also patients who are eligible for this type of testing. Depend on type of the genomic rearrangement you are looking for, you can use different techniques. Different techniques for testing, also looking at the methylation profiles. Whole genome sequencing is commissioned as the clinical test, fully accredited, ISA accredited CMR clinical test for the number of clinical indications. And you know, if you start in whole genome sequencing for cancer patients, my advice is to go first for the pediatric tumors, both hematological, mainly acute leukemias, and as well as solid cancers. Because at least from my experience, pediatric community is the most responsive. This is, well, I hope all oncologists care about their patients, but this level of engagement I didn't see in any other community. Hematological cancers, acute leukemias, you know, they started doing genomic testing probably 30 years earlier than the rest of the cancer community. So there are a lot of genomic markers which correlate with patients' prognosis, diagnosis, treatments. But you know, they're running now dozens of tests. Why don't you replace it with a single genome? We included sarcomas because it's type of cancers which is driven by the big genomic rearrangement, structural variants, copy number changes, which are much easier to spot with the whole genome sequencing. We recently included ovarian cancers and breast cancers, maybe for, mostly for the markers of genomic instability, like homologous recombination deficiency. We also start looking at neurological tumors, And at the moment, all metastatic advanced tumors which exhausted standard of care testing and treatment become eligible for whole genome sequencing. So this is, again, National Test Directory, and this is a slide that I steal from my colleague who is working on colorectal cancer. So if you look at all testing, which is now commissioned by NHS for colorectal cancers, it's quite a list of genomic markers for which targeted therapy is already available. And what is more important, it's different types of genomic rearrangements, small variants, copy number changes, structural variants, and they all picked up by different type of tests. And you know, if you just start thinking about number of testing that need to be done from this tiny biopsy, it's a bit crazy. So if it's colorectal cancer, you want to see if this patient have microsatellite instability, because then patients are likely to respond well to the immunotherapy. So you're doing IHC for Lynch syndrome genes. Then you're looking at small variants in KRAS, BRAF. You're looking at the copy number changes in MET. You're looking at fusions for NTRK. Then you're looking for the tumor mutation burden, for microsatellite instability. FISH and cytogenetics are mainly for hematological tumors, not very relevant for colorectal probably. And then you also looked at the inherited mutations in Lynch syndrome genes, and also what we call now pharmacogenomics, DPYD mutations, which are responsible for the toxicity of the chemotherapy. So this tiny biopsy is supposed to be split between so much different tests, which frequently you run out of the material before you complete your testing. So we see that as a future, whole genome sequencing create this one-stop shop for diagnostic. On the right-hand side, I'm showing what we call circus plot. This is graphical representation of the genome. So now all clinical scientists who make an interpretation of our results, they call it green genome. Because you see this green ribbon, in this case it's also red ribbon. This is density of insertions, deletions, and single nucleotide variants. And you usually, if it's a colorectal patients, you usually see it in the patients with microsatellite instability. This is mean high mutational burden of indels and small nucleotide variants. So it's likely that this patient will respond very well to the immunotherapy. Green genome. Of course, circus plot is not the only thing that we report back to our clinical scientists. I will show some examples later, but you know, this is just so easy to grasp. We all like colorful pictures, figures. So this is what the journey of the cancer genome looks like. Of course, patient is in the center. Staff in genomic labs is recruiting patient, consenting patient, collect sample. For cancers, we collect both tumor and normal sample. Genomic lab extract DNA and then send it to central Illumina facility. So different technologies were mentioned before. Our clinical testing is happening now in Illumina central facility with this short read sequencing, but we're now running pilot with Oxford Nanopore because currently our turnaround times is around 3 weeks for the whole genome sequencing, and of course our target is 5-7 days. Part of the reason that it takes now 3 weeks is because samples are sent around the country. It's not a huge country, but still takes some time. So we thinking about local sequencing next to the patient bed. And with Oxford Nanopore technology, it's much more mobile. The size of the machine, it's not a shoebox, it's like 2 shoeboxes. So you just need bit of bench in the room next to the patient bed, and you can install machine there. And then once, another advantage of the Oxford Nanopore is that you don't need to batch your samples. Like with these big Illumina machines, we do need a central facility, so we can batch samples from around country in a single facility, run it in a single time, so it's much cheaper. With Oxford Nanopore, you can just slide flow cell at any moment. Your patient DNA arrived, you slide it in 2 days, genome is ready. Variant calling and automated interpretation is happening at Genomics England. Sequencing information is sent from Illumina central facility, and now we're also building these networks, pipelines with genomic labs, so we can send this data efficiently between multiple sites. Results are sent back to the genomic labs in NHS, and each genome, results of the genome analysis are reviewed by the clinical scientists. Clinical scientists make assessment of oncogenicity for variants that we prioritized, for both somatic variants and germline variants. If there are some germline findings, then clinical scientists also engage with geneticists. And then these results are presented collectively on the tumor advisory board. And as I said, this is very multidisciplinary. It includes oncologists, pathologists, clinical scientists, geneticists. And then decisions about change to the patient management are made. And yeah, this is how we close this clinical loop. We are very proud that we managed to collect fresh frozen sample. It's still difficult, especially with local hospitals where freezers are not immediately available next to the place where biopsies are collected. So we're looking now for alternative, but here I'm just showing, you know, I spent my first 6 months at Genomics England making this comparison of the fresh frozen with formalin-fixed samples. Formalin-fixed samples is like a standard for pathology labs. So historically, workflows were tuned to collect these formalin-fixed samples. But you know, when you come back to the pathologist and you show them what the damage is done to the DNA, it's pretty convincing. So we managed to do our 100,000 genomes, had like 16,000 cancer samples, and 95% of them were fresh frozen samples. Here I'm just showing the circus plots for the genomes that we generated from fresh frozen and from formalin fixed. Yeah, guess what is where. The central part is showing structural variants. So you can see on the right-hand side, it's very densely populated with these green arches. This is all this cross-linking between different chromosomes. On the circle, I'm showing chromosomes from 1 to 22. So you can see this enormous number of cross-linking, false positive translocations that you observe in formalin-fixed samples. And then you can see this green ribbon and red ribbon. But you know, this is not a patient with very high mutational burden, it's just a patient with high burden of false positives, indels, and single nucleotide variants. And the outmost track is the evenness of coverage of sequencing in the tumor sample, and you can see on the right-hand side, it's very wavy, it's going up and down. It's very hard to call copy number changes in these samples. And look at the left-hand side, it's much less rearrangements. In reality, these patients have very low mutational burden, so all these millions of variants you find in the formalin samples were false one. Okay, I hope you're convinced now that if you want to do genomes, you do it from the fresh frozen samples. We also start looking now at RNA later as a preservative, and we hope that we can keep samples in RNA later for up to 24 hours, and within this time, samples can travel to the freezer before they sent to us. This is an example of results that we returned for all our cancer patients. I picked up this case because it was submitted to us as carcinoma of a non-primary. So looking at the genomic data, can we guess what was the primary for this patient? And tailor patient treatment based on this finding. So it was sent to us as a metastasis in brain tumors. As I said, pathology diagnosis sounds as undetermined cancer. What we found there is p53 mutation. Well, this probably wouldn't help much because p53 is the most mutated gene in tumors. But we also find potential drivers for lung cancers, frame shifts in RB1 and NF1 genes. Then we look at the patient tumor mutational burden. And this is how we are looking at it. On the y-axis is the number of synonymous variants in somatic variants in patient tumor. And because different types of cancers have very different average mutational burden, we presented like each vertical section here is the different tumor type. And you can see that for example, endometrial carcinoma, colorectal cancers, melanomas have very high mutational burden. Hematological cancers or childhood cancers have very low mutation burden. So where was this patient? It has very high mutation burden, above 10 mutations per megabase of sequence. And this is, again, typical for colorectal lung melanoma. And then the third piece of mutation. So you can look at these mutations in specific genes that can be potential drivers, but you can look at the entire set of somatic mutations you find in a patient and look at some patterns, signatures. So for example, in these patients, we saw prevalence of C2A mutations. And you also can look at the nucleotides upstream and downstreams of your SNVs and again look for patterns. So the pattern of mutations for this, sorry, it doesn't look right. So these C2A mutations are usually caused by toxins in smoke. So we call it smoking mutational signature. And again, it's very prevalent in the patients with lung cancer. So this is a conclusion that lung is the likely primary site for this tumor. So patient treatment, chemotherapy treatment can be tailored using this knowledge. But of course, we are not doing genomes just to look at the small variants. And you know, this is another example. So this was a patient with ovarian tumor. And we find here the CDK12 mutation. And we have now this system, we call it SVIG. It's a scoring system for somatic variant interpretation. It was developed by our clinical scientists. So looking at different parameters that characterize this mutation, you can make a classification and say whether it's oncogenic or benign. But you know, you can look just at this small variant, but you can also look at the, again, the signatures, like what's the consequence of this single mutation. And again, circus plot on the right-hand side. And probably even without much knowledge, you can see very specific patterns here. You can see a lot of these blue arches. So blue arches are tandem duplications. This is a type of instability which is known to be driven by the deficiency in CDK12. So when you pool these different types of mutations, different strain of evidence, you can very confidently say that the CDK12 is a driver in this case, and this is drive this instability in the genome I think genomic instabilities, different types of genomic instabilities, is a very hot topic. Last 5-10 years in cancer genomic research. It's not quite made its way into clinical practice. Homologous recombination deficiency is, of course, kind of pioneering in the clinical practice. At least in ovarian cancer, it's already a marker of response to the PARP inhibitor. But I am sure in next 5-10 years, we will see much more treatments become available for different types of instabilities. And I think this is where the strength of the whole genome sequencing. It's not just this identification of small variants in drivers or identification of specific fusions. It's rather these patterns that you can see in genomes that you wouldn't be able to see in the panel. You know, for me, panel is like when you mask genomic information with an untransparent, I don't know, piece of paper, and then you cut these small windows. So you just see little bits of information, but you're missing the big picture. Once you have whole genome, you can look for these patterns. And in the future, we probably can use these patterns. We can cluster our genomic space into the groups of patients with different types of genomes, different types of instabilities, which developed in tumors. And then because all our genomic data is linked with clinical data, we know how, like what was diagnosis for patient, how it was treated, how it's respond, what was survival for patient. We can assign new patients to these clusters and then make decisions about patient treatment based on the clinical information that we already collected for these clusters. So we see, oh, this cluster of patients respond very well to this specific treatment. Probably our new patient will also benefit from it. So this is like a future thinking for how we can use genomes in the cancer space. I already mentioned that we're doing this Oxford Nanopore pilot. And the main advantage is that this is a very mobile technology. But of course, coverage across genome is much more even, including repetitive segments, regions of segmental duplication. I'm giving here a few examples from the RANKSOX1 fusion. There is this nasty 250 base pair 80 reach repeat. It doesn't sequence with Illumina 150 base pairs reads very well. In the bottom is Illumina sequence, and you can see the coverage track. It's very wavy coverage. At the top is ONT, and you can see much like square patterns. So coverage is even. In this case, we missed a breakpoint for this fusion in the Illumina data. We picked it up very efficiently from the Oxford Nanopore. Better resolution of complex structural variants. And you know, with Illumina, we missed all fusions in rearrangement in centromeric telomeric regions. Of course, we can pick them up with ONT. We can phase breakpoints. Very importantly, you can look at the genomic variants. You also can look at the methylation profiles. This is my new love. The resolution of the methylation signals is amazing. This is just the last slide for the genomic medicine services. This is actionability data that we collected from our genomic labs. I present them here in 5 groups, depend on the patient clinical indications. And you can start seeing very, well, it's a predicted patterns like for sarcoma, you can see that most of actionability come from structural variants, copy number changes, while in other tumor types, probably small variants are like in adult tumors, small variants are more informative, more actionable. But you can see that across the board, for clinical indications for which we're doing now whole genome sequencing, we find actionable mutations in more than 50% of the patients. And we're doing now in parallel panels, genomes. In 10 to 20% of the patients, we have new actionable findings with whole genome sequencing compared with panels and all our standard of care data, which is done. Okay, I have a small bit about our historical data that we generated as the 100,000 Genomes project. As I said, 16,000 cancer patients were sequenced as a part of this project. All this data now sits in our research environment. I'm giving here a link for how to join we called it Genomics England Cancer Interpretation Partners. So all this data is publicly available. Your institution have to sign agreement with us and you get access to all these 100,000 genomes. Our manuscript describing data is now in press in Nature Medicine, so hopefully you will see it in a few weeks. So most of this is just figures that I stolen from the paper. This is just to show you how 100,000 genomes was covering patients across the country. It was not just from the big hospitals. It was from across the country. Those are the tumor types that we sequenced. The most representative one were breast, colorectal, renal, and very importantly, we have a big collection of, and very diverse collection of sarcoma genomes, if it's part of your research. This is a prevalence of the different type of mutations, or you can call it landscape of mutations, that we find in this cohort. So we mapped variants that we've seen in these 16,000 genomes. Actually, it was 13,300 something, because we put aside now hematological cancers and pediatric cancers. They will be subject to separate publications. So this is adult solid tumors, and this is prevalence of mutations in the genes which are already in our test directory. Actionable genes for which there are therapies or prognostic diagnostic markers also included. And you can see that like these vertical sections for the small variants, CNV, structural variants, pangenomic markers like homologous recombination deficiency or high mutational burden, and then also genomic predisposition. So the selection of clinical indications which were included as clinical testing in the test directory was based on the prevalence of these mutations. For example, I mentioned that we're doing now whole genome sequencing for brain tumors, and this is why, because there is a high prevalence of actionable mutations in small variants and also copy numbers. We are doing whole genomes for ovarian patients, because again, 40% of these patients have homologous recombination deficiency and eligible for the PARP inhibitor. Also, this population have high prevalence of genomic predisposition, which probably have in sequence for patient family management. I mentioned that we're doing whole genome sequencing for all sarcoma patients, and this is why, because they have high prevalence of actionable mutations and copy number changes and fusions. So this is how we picked up clinical indications for our genomic medicine services eligible for whole genome sequencing. This is also a very important message for our pharma partners. I highlighted here the case of the PIK3CA mutations, and patients with PIK3CA mutations already eligible for treatment for PIK3CA inhibitor breast cancer, but you also can see a high prevalence of mutations in other clinical indications. For example, ovarian endometrioid adenocarcinoma or uterine carcinomas. So those are the clinical indications where potentially pharma companies can extend their clinical trials to. So this, as I mentioned, all this data now sits in our research environment, but very importantly, it's also linked with the wealth of clinical data. And you know, this is not like a clinical trial which is followed up for 5 years. This is lifelong follow-up. And thanks to our national health system, we have like clinical data for this. We already have this clinical data in a centralized structure, so we don't need to reinvent the wheel. And we have data on diagnosis, on all the hospital episodes which happen with patient, treatments, response, and survival. And you know, this is just one of the examples of what you can do with this wealth of data. This is survival analysis, and it's a part of the Nature Medicine paper. You will see much more information there. So this is survival for patients with PIK3CA mutation. So using data in the research environment, you can say that patients with PIK3CA across different types of cancers have better survival than the wild type. And you know, this data was already published for some specific groups of the breast cancers. But we also find that this is, it reproduces itself across different types of cancers. Like this is where we extend in, so I already mentioned our pilot using Oxford Nanopore technology. We also merged now our 100,000 genome project with digital pathology images, so we can combine clinical data, image data, genomic data, and also with radiology images prospectively. And you know, this is just for the conclusion, so I talked about our healthcare loop, and how we deliver now whole genome sequencing as accredited test for our genomic medicine services. Then I talked about our 100,000 genome data, which is sit now in research environment, available for basic research for pharma companies, for as a source of knowledge for development new treatments. And you know, this is how we see it as an infinity loop. Data that we collected for clinical use, and of course it's all consented, patient can opt in research option, making their data available for the research. And by the way, from our experience, 95% of the patients tick this box and make their data available for research. Data flow to the research environment. Well, unfortunately, we are not covering your project like Qatar is doing, but you know, it's a huge amount of data. You don't need compute, you're just using our compute to generate new insights. If they're translatable into the clinic, then we are there to talk with you, to discuss the value, and we already have quite a few collaborations with academic groups. When we have a problem, there is no bioinformatics problem, analytical problem. We don't have available algorithm for it. So we outreach to our collaborators, develop software together, test it, validate it, accredit it, put it into our clinical pipeline, and we find it's very efficient, very beneficial for our patients. And yeah, I just want to thank our collaborators from the research, our collaborators from our health system, clinical team from Genomics England, and of course, cancer analysis team. Thank you very much.